Okay, good afternoon. Can you hear me?
Yes.
Okay, I have three questions regarding the new topic, fifth lecture. So the first question is, you've said that the centrosoma is very important, how to say, the organelle, yes, very important organelle in mitosis. Also, only animal cells contain this kind of organelle, but not plants. So we know that the reproduction of plants are helped by mitosis, and how they can just pull to polars if they don't have any centrosomes. I mean, pulling the DNA.
Yeah, I understand it. Okay. So is the only question? You say three questions?
Oh, okay. The second question is, you also said that microtubules help to intercellular transport. What connection? And I was wondering what connection they have between this transport and to this, to his structure, helix structure. So it's like protecting this some organelles to transport to another side of cell, or how? So I a bit couldn't understand this part. And the last question is, can the dysfunction of centrosome lead to Down syndrome? Because when it pulls the wait, how to say 46 46 chromosomes to each side. So, for example, dysfunction of centrosome can pull 47 chromosomes, and to other side 45. So this 45 chromosome cell will die. Then the reproduction of this mutation cell can lead to Down syndrome. Is it possible?
Okay.
Dysfunction of centrosome.
Mm-hmm. So I'm sharing the screen for now. So do you see my screen?
Yes, yeah, we can see.
Okay, nice. So the first question was about the centrosome in mitosis, and let's combine them with the third question about the Down syndrome. So first of all, you have this centrosome. So let's say in mitosis, you have divided centrosomes, so they form mitotic spindles on both polars of the cell, okay? And whenever we have the doubled set of DNA, they start pulling them. And this makes this DNA move or segregate. And thus we can then divide the cell into two equal parts. And both cells, since they are the same, they will be living cells that are kind of ready to divide and live their own lives or their own fates, so-called. Okay, so this is about the mitotic spindles and how they are related to centrosomes. Okay, so the centrosome in plants, it doesn't exist, but they have their own organelles that are responsible for this segregation. And these organelles, so I'm not really sure the name of plant segregation complexes or something like that. And I will need to look it up, and if I find the information that will be easily accessible, I will share it with you, okay? But the thing is, the centrosome itself was developed in Animalia, so starting from protozoa, and this particular organelle is only the characteristics of Animalia. Okay, so it's everything that moves. So birds, humans, fish, etc.
Mm-hmm.
So this is about the centrosome. And now the Down syndrome. Of course, whenever we have the disruptions in the process of this segregation, we might end up with one cell having, let's say, 1/3 of all doubled DNA, and the other part will have 2/3. Okay. In this case, we will have a big problem for both of these cells because both of them will live through stress. In the first case, we won't have enough genes to maintain cell stability and DNA integrity or stability too. And in the second part, in the second case, we will have too many genes that will try to compete between each other. And this will lead to starving of the cell because all of these genes will start expressing and consuming the nutrients. So this is one of the ways that this cell dies. Another way is just apoptosis. So it's a programmed cell death. So whenever we have too many DNA molecules, the cell might kind of tell itself that it needs to die, so it goes controlled suicide and just dies. Okay. In some cases, like in Down syndrome, for example, we have this segregation problem on the level of zygote or even before that. So whenever spermatozoa or oocyte, so during the development, we might have these segregation problems, and this will lead to an additional chromosome in one part. Okay. So we have plus chromosome here, and here we have minus chromosome. So these for sure will die. And since in Down syndrome, this plus chromosome is kind of fine, so this cell still can live. Not as good as usual cells, but it's still viable, okay? And in this case, in the adult organism, so whenever we have like fusion of spermatozoid and oocyte, we will have one additional chromosome, and this cell will be viable, which means it can live, okay? And since it is viable, we kind of keep this mutation, and the development just continues. Development. Okay, I hope it was clear for Down syndrome and microtubules and the intercellular transport. So I'm going to delete this screen, and I really hope that you all understood. Just a second. Fariza, so I will answer this question afterwards, okay? So we have the intercellular transport. So the helix of these monomers, so as you remember, the microtubules are formed like this. So they are forming this huge polymer. And the thing is, well, the characteristic or the trait of this hollow cylinder doesn't do much. All you need to know is that we have minus end and plus end. And some proteins, they kind of use this microtubule as the road, okay? So assume these adenine and kinesin, some kind of cars that are moving on top of the road. And this road is your microtubule. So this is microtubule, and let's call it the road or the highway. For motor proteins. And motor proteins are cars. And since we have a lot of these microtubules inside the cell, this will mean that, for example, we have a mitochondria here, and it is attached to this motor protein, and it starts moving, and we need to get it here, for example, in this place. So it starts moving on top of this microtubule, and using this mesh of microtubules, it gets to the place. And in this case, our mitochondrion is a cargo, and it is delivered to a destination place, okay? And I hope that microtubule transportation is also clear. So, who was asking the question? Sorry.
Yeah, I am. So yes, about microtubules. Okay, I have understand that. So the last question. Now, can we say that dysfunction of centrosome, like causes of Down syndrome, also considered as the dysfunction of centrosome, or not?
Again, the question, please.
Can we say that one of the causes of Down syndrome is dysfunction of centrosome?
There might be many different causes for the disruption in the segregation. Even the change in pH of the cell can lead to this spontaneous disassembly of these microtubules and actin filaments. And in this case, the whole process of segregation kind of stucks. And at some point, it might bring some chromosomal aberrations and chromosomal anomalies. So also what we have, for example, the increase or decrease in temperature. So the usual temperature for cells is about 37 degrees. And if we have temperature higher than that or lower than that, it will also lead to either increase of chemical reactions or decrease of the velocity of the chemical reactions. And this will also disrupt the fine process of this segregation. So anything that you could imagine, as well as free radicals, for example, they can lead to these segregation problems. Okay, so there is no single defined cause or reason for the problems with segregation. Okay.
Okay, thank you.
So the question from Fariza. So Down syndrome is actually happening. I mean, this problem of segregation is happening on the stage of the development of oocytes, as far as I remember. So the problem with this oocyte, so one part, I mean, the oocyte that was a sister to the one with 24 chromosomes, it dies out, okay? And we have one oocyte with excess or plus chromosome. And after we add genetic information from spermatozoid, for example, we have 47 chromosomes in total, and the child kind of develops this Down syndrome, okay?
I'm sorry, I just, as far as I'm concerned, Down syndrome is due to the dysfunction of segregation in 21st chromosome. So it's not a sex cell, it's a somatic cell, and it's just what I remember from school, so that's why I said it.
So it's actually
Somatic cell.
It's actually the topic of tomorrow's lecture. So do you see my screen?
Yes.
Okay. So we have 22 chromosomes that are called somatic, and one chromosome pair, or let's call it just chromosome. So two chromosomes, which are X and Y, they are sex chromosomes. They are not the same as somatic cells and sex cells, okay? So the somatic cells, they are diploid, which means they have 46 chromosomes, and two of them will be either XX or XY, so male and female. And for sex cells, you will have 23 haploid cells. No. So it's 23 chromosomes in them, okay? So 22 somatic and either X or Y. So these guys will be the 23rd. But this is not the same as this, okay?
Hello.
Hello.
I have one question regarding actin filaments. So
Okay.
I didn't understand actin filaments at all, like all this lamellipodium and other things.
Okay, just a second.
Okay, I just don't have and I can't imagine like how do they work and so on.
Just a second. Do you see actin filaments now?
Yes.
Okay, nice. So first of all, again, we have, just a second. Okay, we have these monomers, and they build this filament, which is the polymer, okay? And this polymer plays role in the formation of such elements as filopodia, lamellipodia, microvilli, so these guys, and also stress fibers. So what are the composition of these structures? They are usually formed mostly of these actin filaments, and they are needed to move some parts of the cell. And for example, if you take these lamellipodia and filopodia, they are kind of structures that are always close to each other. So for example, if you take this cell, it starts kind of making these protrusions or filopodia. So they move in this direction, all of them. So just imagine first cell to be like this. And then it starts growing this filopodia, okay? And it builds it through the growing of these filaments towards the edge of the cell, okay? And whenever it starts growing this filopodia, the cell surface is kind of increased towards this end, okay? And when we have enough filopodia, they start kind of broadening towards each other. So it usually looks like this. And this thing would be called lamellipodia. And this everything is filled with actin filaments. And this means that the content of the cells start moving towards this direction. So in this direction. Okay? Is this part clear?
Yes, I get it.
Okay,
Like
so
it
what?
expands, the cell expands and like moves towards one direction, right?
Yes, that's right. So it extends these fingers and then builds some kind of connections between these fingers, and these connections are lamellipodia.
Oh, I got it.
Okay, nice. That's how it actually moves from one part to another. So what are microvilli? So one thing here is also a protrusion, but if you think about the cell, it stays on the surface, okay? This is some surface. And this is the basal part of the cell. And you need to kind of, just a second. Okay, so here you have some kind of surface. And here you have another cell, and also here. So these microvilli, it is for kind of uptaking stuff from the outside. So let's call these, let's assume that we have some kind of nutrients here. And since the cell can kind of sense these nutrients because it has sensing apparatus or receptors on its surface, it starts growing these microvilli to kind of get these nutrients and uptake it. So it grows them, and as a vacuole, it uptakes it inside the cytoplasm. And then it goes into the digestion inside the cell. So we have proteolysis, for example, or lipolysis. Just a second. Okay, so it's another question. And these microvilli, they are also moving. Oops. They are also moving towards something. Okay, so whenever you hear about movement, you should think about the actin filaments. And these actin filaments, they kind of move towards these nutrients. Is it clear here?
Yes, it is. Thank you. I understand.
Okay. And finally, we have the stress fibers. So just imagine, okay, so again, imagine some kind of needle here, and you try to poke this cell. So it rapidly grows these stress fibers. And after that, across these stress fibers, these cell starts contracting. So it contracts, trying to avoid this needle because it senses the danger from here. So this part is not fine with this needle. And since it starts contracting, finally we get something like that. So the cell changes its shape, okay? If it doesn't move to another place, it at least starts changing the shape. So we avoided the problem. Is this part clear?
Yes.
And finally here, for example, in the contractile ring, so you have a cell and you successfully segregated chromosomes, so they are on different sides of the cell. But now you need to really divide this cell here. And it starts growing these actin filaments that will kind of build the ring. And then this ring starts contracting. It contracts till the time that these two parts, I mean the cell 1 and 2, they are being split because of very thin cytoplasm here. And this is the function of the contractile ring. Is this part clear?
Yes, it is.
And finally, we have cell cortex. So cell cortex is everything that kind of keeps the cell shape. And you can also see microtubules here. So this is the real carcass or exoskeleton, let's call it exoskeleton of the cell. So this shape, if we don't have this cortex, the cell would just kind of, how you say that, extend in all directions, because if you think about the drop, so if you have a drop of one size and nothing keeps it in the place, it starts moving in all directions. And finally, we have just a spill, okay? In this case, this cell will maintain its shape, and this cell also will maintain its shape. And even if some part of it wants to kind of protrude or extend to this direction, this carcass will not let it do that, and you will have just a constant shape of the cell. So this will not happen, and the cell shape will stay the same. And the question from Abayhan. So on the slide with microtubule cell division, I'm going to be curing cancer. No. The problem is it occurs, the anomalies of mitotic spindle and centrosome occur in cancer and genetical disorders. Abayhan, if you look at this slide, so here you can see centrosome and mitotic spindle anomalies, and this is a problem. Usually, everything that is related to centrosome or mitotic spindle is controlled, so it's a very tight control. Controlled. If anything goes wrong during the formation of centrosome or mitotic spindle, the cell would prefer to commit suicide. Okay, and this means that whatever happens to mitotic spindle or the centrosome, it is risky. So the problem is risky, and cell really is on the edge to go this way. Okay, but in cancers and inherited disorders, these anomalies are skipped. So instead of going to apoptosis, the cell will still divide with the problem. Okay, so we have a question about 5 types of intermediate filaments. Okay, so in intermediate filaments, we have 5 types, and first 2 types are acidic, so it's type 1, and basic keratin, and they both form this road shape. So one is acidic, another is basic keratin. So whenever they bind, they form this keratin filament. And keratin filaments, they form junctions between cells. Okay, so they are responsible for stability of tissues, for example, or cell interactions. So the keratins themselves are very important for the cell. So if you don't have these cell junctions or anchors to the matrix, you will have a lot of problems. So these are type 1 and type 2 filaments, intermediate filaments. Type 3 is responsible for first transmitting membrane receptor signals, also fixing of some organelles inside the cytoplasm, and also supporting cell membrane. So you have this cell shape, and whatever happens inside the cell, the shape is almost the same or very constant. So the same might happen for the organelles, let's say mitochondria, and assume that at some in some cell type you have a huge concentration of this mitochondria in one side of the cell, and it really needs it there because it produces a lot of proteins, for example. And if we don't fix these cells, they start floating and create these regions. So let's say that a few of these mitochondria, they float away. So we don't have few of them. But because we have the region that produces a lot of proteins here, the energy very rapidly goes down. So the capability of the cell to produce proteins drops. So protein production is decreased too. That means that we really need to somehow fix the organelles so they don't float around. Now we fix it with intermediate filaments such as desmin, the GFA, the peripherin, and vimentin. And this is for fixing, and finally the transmission of the membrane receptor signals. So sometimes one part of the cell is responsible for receiving the signals from the outside, but the activation happens on the other side. So let's say it receives the signal to detach from the surface. So here is surface. We need to detach the cell from the surface. How do we do that? We transmit the signal across these intermediate filaments. So they, let's say they change the structure, and this change kind of goes through all of these filaments, and when it reaches this part, here is the sensing molecule that understands that the change of shape of this microfilament happened, intermediate filament. And since it senses this change of shape, it also starts producing proteins that detach the cell from the cell surface, from the surface. And whenever it happens, we have the response to this ligand or this signaling molecule. And this cell took this signal from the upper side, but transmitted it to lower part, okay? So we have type 4 intermediate filaments, and they are neural ones. So they are responsible for maintaining shape and diameter of, for example, axon. And this is the neuron. And all neurons will have one axon. a single axon, and the bigger diameter of this axon, the more processes we can maintain inside it. And it means that we can have faster signal that goes through this axon. And if we have faster and safer signal, it means the response is very related to the diameter. So, okay, so it influences the neural functions. Finally, we have the fifth intermediate filament type, and it is related to this nucleus. It really makes it very firm and rigid. So if we have some kind of shakes or quake for the cell, the DNA inside it will not leave the nucleus and will not float outside the nucleus. Or nothing will enter this nucleus. So let's say we have a nucleus with the very firm borders. And these very firm borders, they are kind of control points that do not allow free access of outer agents to the DNA. At the same time, they don't allow the DNA to leave the nucleus. These are lamins. So we have intracellular transport, and again we have a cell. And let's say we have one superspecific organelle in one part of the cell, and we really need to transport it to another part. So first of all, the cell is full of these filaments that form some kind of mesh. And the motor proteins, they use these filaments as the road. Okay, so filaments are the road for motor proteins, as if they were cars. So one motor protein, it takes this mitochondrion and starts moving across these filament, and it moves to the destination place. Okay, so let's say this was the destination, and whenever it moves, it uses ATP. And since we have energy, since we have movement, we need energy to fuel it. So ATP is kind of gasoline. So in this case, we have this intracellular transport, and not only organelles, but also nutrients, and usually nutrients are inside the vesicles. And these vesicles are usually something that is happening due to endocytosis. So let's say here we have the nutrient, and then these two parts, they kind of bring it inside, and we obtain the vesicle with the nutrient. And this vesicle is attached to the motor protein, and this motor protein, it starts moving across along these microfilaments and gets to the destination place. Again, using ATP. Don't forget about the ATP. And I hope it was clear.